Okay, so last time we looked at how ligand binding affects the saturation of a protein. We were able to quantitate that. We derived the binding of that um, ligand to the protein and figured out that it was a function of ligand concentration, at least at a given um, concentration of protein. And we determined that the KD um, is a measure that's specific to the particular protein ligand interaction. And so we can use this KD to get some information about the nature of the protein. And so here are various protein ligand interactions. Make this a little bit brighter. And the KDs that correspond to them, right? And what you can see is that the the smaller the KD, the more, the tighter the reaction, the stronger the affinity of the protein for the ligand is. And so the kind of tightest one that we see is the biotin avidin relationship, and this is actually exploited in a lot of molecular biology um, uh, techniques looking at this tight relationship. So this KD is at 10 to the minus 15, which gets into, I can't even describe what that is. Nana is 9. What is it? Pico is 12. So I don't know what 15 is, but it's pretty freaking tight, right? Versus some others that are in the 10 to 7 or in the micromolar range, right? And we can look at various types of protein ligand interactions and see what, in general, their range is. So, an antibody antigen interaction, remember, antibodies are designed for antigens, are in the 10 to the minus 8 to 10 to the minus 12 range. Protein ligand DNA ligand, DNA protein interactions are roughly in that same range. Receptor ligand interactions, kind of the hallmark of neurochemistry and all sorts of biology, is around 10 to the 5 to 10 to the 10. And note that enzyme substrate interactions tend to be weaker than most other protein ligand interactions. It is a subset of protein ligand interactions, enzyme substrate interactions, but they are of lower affinity. And we will discuss why that is. There is a reason for that, that has to do with the transition state theory that we will talk about when we talk about enzyme kinetics. But make a note of this, enzyme substrate interactions tend to be of lower affinity than most other protein ligand interactions. Again, all of these are reversible. All of these are probably, for the most part, non-covalent, right? But they have different KDs, and because of that, different affinities. So now we're going to talk about two proteins in particular. And again, I stress every biochemist talks about these proteins in terms of protein function, but there's nothing really special about it except that, you know, it is what we started with. It's a good example of protein ligand interaction. It's a good example of how the structure of a protein really corresponds to its function and what that function is. But there are thousands of other proteins that we could use. And as I've said, alluded to before, I am tempted to use other proteins to do the same thing. But then when you all get to medical school, graduate school, and they pop up their picture of hemoglobin, you would come to Dr. Sharif and be like, Dr. Sharif, how come you never told me what hemoglobin looked like? And y'all would be mad at me, right? And you wouldn't donate to Morehouse College, and the president will fire me, and it will be a whole thing. So I'm gonna do what we do. Maybe a little bit later on in the week, we'll do some things related to COVID that looks at protein structure as well. But we're going to start with an analysis of myoglobin and hemoglobin. And these, obviously, you can probably tell by their name, globins, they're related proteins, but they have different functions. Myoglobin stores oxygen. It's in the muscle, and it only releases that oxygen when there is a crisis. So when those muscles have depleted all of their oxygen, then myoglobin will step in and give a little bit more. Hemoglobin, on the flip side, is predominantly in the blood, and it is used to transport oxygen from the lungs to the cells. So it has a much looser relationship with that oxygen because instead of having it be kind of bank of last resort as it is with myoglobin, this is supposed to freely give out that oxygen just as it's passing through the particular cells. And there's a really ingenious mechanism to do so so that it can hold on to some oxygen for those places in the body where you really need it, but yet release some for general function. And we'll talk about that in a minute. So hemoglobin actually transports oxygen from the lungs and brings it to your cells throughout the body. Right? Both of them work the same way. They bind protein, they bind the oxygens reversibly using an iron. And I'm not going to get too much into metal chemistry here, but it is a iron in this divalent state that is associated with the porphyrin ring that is bound to the protein of the heme group. One thing that's interesting is, if you remember from general chemistry, iron has multiple states. If this was iron 3 plus, which is certainly possible, it would bind that oxygen irreversibly and be useless for its function. So this is a look of the structure of myoglobin and hemoglobin. Important to note, myoglobin is a single polypeptide, so this would have a tertiary structure, but not a quaternary structure, right? Whereas hemoglobin is four polypeptides, so it has a quaternary structure associated with it. Now, hemoglobin is what is known as a heterotetramer. There's four different subunits. It is also a dimer. This is a dimer would be two identical subunits. And so here they have an alpha and a beta subunit, and those are mirrored on either side. So here alpha subunit, beta subunit, right? And we don't get into symmetry here, but it has a multifold level of symmetry. That would be the topic of an advanced biochemistry, or really a protein biochemistry course. Okay, so both myoglobin and hemoglobin bind to the heme group that we talked about earlier. Hemoglobin has four heme binding sites. Let me make this brighter. Four heme binding sites. And each of those are able to bind a molecule of molecular oxygen. So hemoglobin can bind a total of four molecules of oxygen, whereas myoglobin, because it only has one heme and one subunit, can only bind one molecule of oxygen. And if we were to compare these two, we would see that the myoglobin looks like both the beta subunit of hemoglobin and the alpha subunit of hemoglobin. I think I have that here. Yep, good. And so note for both of these, so this is the myoglobin polypeptide, which is a complete protein, and this is one subunit of the hemoglobin over here, and both of them look fairly similar, both full of alpha helices, right? Both kind of surround the heme group and kind of cradle it. You can see some differences. Note here, this alpha helix has one, two, three turns. This one has one, two turns, right? This alpha helix is much longer than this, but essentially we can look at it and see the similarities between these two structures. Both of them grab on to this heme group and use that ability to grab that heme group in order to bind molecular oxygen. So we'll go into the details of that in the next go round.